So this is the second of a series of 3 talks about electro-diffusion applied in dendritic spine, and today we'll discuss the aspect of modeling and numerical simulations. So all softwares are available on bionumetrics.org at the following address, and some of the content was already published in this neuron paper in 2018. So I would like first to remind you the context of this study. So we were interested in dendritic spine, which are those small protrusions located on the dendrite, and you can see here more specifically in green, yellowish, you see this the presence of a spine apparatus, which is an organelle located in this structure. And so if we go to the next slide, next figure, we see here an electron microscopy picture of a dendritic spine, where usually a current flows through channels here located on the surface of this postsynaptic density. And what we are interested in is the following question: can we estimate the difference of voltage between 2 points, let's say point A here and point B here? So if we are capable of calculating this, then we will be able to calculate any difference of potential on this dendritic spine when a current is flowing inside. So let me summarize the 2 questions. How to compute the difference of voltage VA minus VB, and how this difference of voltage depend on the geometry and the input current I. So this is just to remind you the context of this calculation. So there is a difference in ionic concentration between the inside the cell and outside, in potassium, sodium, calcium and chloride. And if you look at those numbers here, if you compare them, you see clearly that in terms of ion structure there is no electroneutrality. That is, there is not enough chloride charges to compensate. And this is probably, there is probably some compensation due to the negative charges located on protein, but it is very much less known, and it's the documentation on this is very poor. However, to maintain these gradients, there are pumps, ATP pump that consume energy, that are capable of maintaining the difference of concentration. For the information, we have developed a couple of years ago a model about potassium recycling, which involve actually glial cells, and this is in this following paper in PLOS Computational Biology, published in 2015. So although it's not going to be the major focus of this talk, I would like to insist that in general there is no electroneutrality at steady state from ionic species, and it is unclear what is the length scale of electroneutrality. That is, what is the scale for which at which you are sure that the number of negative charge is of the order of positive charge and with low fluctuations. And we think it's much larger than the Debye length, much larger than just few nanometer. It could be tens to 100 nanometer. This has to be confirmed. So now the question, if we want to calculate the voltage, we have first to start at a certain level and to understand how ionic fluxes are regulated. So the best way to regulate fluxes is through these ionic channels. However, the model for this is unclear. If we start at a molecular level, then we need to go to the classical Langevin equation, which says that the acceleration plus friction plus forces equals the random fluctuation due to Brownian motion. So the difficulty in this equation, there are 2 terms. that are, that could be not easy to handle is the acceleration, but in the case of large friction, actually the acceleration can be neglected. This is called the Smoluchowski limit. But then the major difficulty is to calculate the electrical field generated by all the moving charges. So this stochastic description leads to the following ensemble of equation. If we want to follow each particle, each ions, then the velocity equals the forces plus the Brownian the friction, the Brownian fluctuation forces. And then this equation has to be coupled to the Poisson equation, which is an equation for the voltage. So epsilon is a permittivity, epsilon 0 is a permittivity, epsilon is a permittivity when we're in water. So epsilon 0 is the divergence of this, equal the charge at the location of each ions. And this leads to a very complicated system where we need to solve an ensemble of stochastic equation and at the same time this partial differential equation. So this approach based on Monte Carlo simulation is in general very hard to implement due to the charge number and the large variety of particles, except for ionic channels. And I will refer to the work of Eisenberg and Schuss, where the calculations have been done and simulations were used to look at how channels can conduct, some specific channel can conduct ions. And this is possible here because the geometry of a channel is extremely simple. At least it can be approximated as a cylinder. So the description at the level of one charge, the Langevin equation, which is summarized here, can be replaced by what is called an equation for the density of charge, that is the number of charge per unit of space, and the derivative with respect to time equals the diffusion term plus the drift term due to the electric field. So this is called the Nernst-Planck equation, which has to be coupled with the Poisson equation when we have here an ensemble of positive charges and here an ensemble of negative charges. And this epsilon account here for the permittivity, which account for the effect of water on the ions. So to summarize, we have two species, then we have the Poisson equations to calculate the field for the moving charge plus possibly some fixed charge with a certain density rho. And then we need to solve the two Fokker-Planck equation for the positive charge here and for the negative charge, where we have a diffusion term in both equations and the drift term due to the generated electric field. And the electric field is generated due to the possible difference of concentration between positive and negative, which is expected when a current, let's say a current of sodium ion flows inside a dendritic spine through channels. So now I would like to summarize the framework for the simulation. So modeling electro-diffusion in a dendritic spine shape. So we define the shape geometry as a ball connected by a narrow neck to the dendrite. So the current is injected through a small narrow disc of radius 10 nanometer, which represents an average, what is called in mathematics in the homogenization of the boundary conditions for entering current. Then we can always ground the voltage here to 0 because the voltage is defined up to a constant, so it's always possible to add or to subtract any constant of interest. Actually, we are interested in the difference of potential between 2 points, so any constant here will disappear in this process. So the radius of the neck is about 100 nanometer. So we have already discussed this system of partial differential equation, and at steady state the derivative with respect to time is equal to 0. Now what I would like to discuss are the boundary conditions. So what are boundary conditions for the different part of the spine that we have to numerize? So first of all, there are these two boundary conditions that are for the positive and negative charges that are going to be defined and given here at the connection between the dendrite, so here is the dendrite, and the spine neck. So because the dendrite is much bigger than the spine, this says that we connect the concentration to the large concentration that is maintained in the dendrite. So we already said that the voltage can always be grounded at 0, otherwise we can add or subtract a constant. Then the negative charges, the fluxes of negative charges at the entrance here, I, so this is at the entrance, so this is here, this place which is called omega I. So we said that it is 0, that there is no flux of negative charges. All right. However, there is a flux of positive charges, which is equals the injected current divided here by the surface of the area. F is a Faraday constant, and D is the diffusion constant of the positive charges that is injected. Everywhere else, so this means on the reflecting part, for example here, d omega r, everywhere else we impose that the concentration, there is no flux of particles, which means this dCp dn and actually dCp dm here equals 0. And also we impose that dVdn equals 0 on this two region, on the reflecting part and the injecting part. So this is the ensemble of boundary condition that we have added to simulate the effect or to predict the difference of voltage when a current is injecting. In another YouTube, I'll probably spend more time to explain that this ensemble of conditions are actually suitable, although some of them could be changed if we wish to, for example, incorporate the effect of the membrane. But this effect is actually negligible, and this is what we have shown in our in the supplementary information of the neuron paper I presented before. So now, how to implement this partial differential equation? Then we need to make to use a discretization of the domain using a mesh grid, and you can see here all of these little dots are an example of mesh discretization where you can see here close to the boundary you have a refinement of the mesh, as you can see here when there is no neck, and also at the entrance, while if you are here in the middle the grid is much more sparse. And so we use this grid to solve actually the partial differential equations and to calculate the field at each point of this mesh. So the first example is going to be the case of a dendritic spine with no neck. It's like having a very short spine, and we have already discussed the different numbers for the geometry. So what we found is that when we inject a current of about 150 picoampere, what you can see is something which is quite interesting. So we looked at the solution along this axis here, the blue axis, from 0 to R, where R is the radius of the spine head. And so what we found is we found three regions. A region here where you can see that the voltage is a large drop of the voltage in a region of about 100, let's say 100, 200 nanometer, which is quite interesting. Which says that there is a region which is called, in the mathematical terminology, a boundary layer, a layer where you have a large change in the voltage. Then once you enter, so probably around this area, you have a region where the voltage is almost constant. And then finally another boundary layer in the third region, so let's say region 1. This is going to be region 2, and this is going to be region 3. So this is 3, this is 1, and the rest is 2. And so you can see here again, although it's less pronounced than in region 1, you can see how the voltage is changing when the charges are going to be extruded, or where they can leave from the spine neck. So the voltage is almost constant except at the entrance and the exit of the dendritic spine. And again, these lengths here are much larger than the Debye length, suggesting that the current can be felt deeply inside the spine head. Then we repeated the similar simulation, but this time we added a spine neck. And so we have already discussed in the previous slide this case where we are inside the head. And you can see here what is the effect of adding the neck. And so basically it creates a large difference of potential. You see how what's the drop here, for example, for the case where a current of 300 picoampere is injected. So again, we have a first region where you have a large drop. Here you are in the head, and then here there is a connection, and it's not a straight line, but there is some deviation, small deviation. But what is interesting is the following aspect about the geometry. If we have a length of a spine neck of about 1 micron, and if we inject about 300 picoampere, then if we reduce, for example, we inject a smaller current of about, let's say, 150, but we reduce at the same time the spine neck length, then we see that it's not just a scaling. There is some clear differences between the difference of potential here, between those 2 points, and the difference of potential between, here I should maybe change color so it becomes clear, between, I should have drawn this in, let me do it again. I should have drawn this in red actually. So if the difference of potential between these 2 points here compare to the difference of potential here between this one and that one. Okay, so we think that there is no compensation by, even if we have a smaller current, if we reduce the spine neck, it's not just a scaling. So there are effects here of the geometry which probably are very interesting to further understand. So in addition with this methodology, what we can have is really we can have access to the difference of potential, and especially here, so let me point for you this case where we inject the current just at the North Pole, so we have a magnification here, and you can see through this voltage line how specifically the voltage is changing. So it is very large here, although from here to here, the difference is quite small, and then it decays almost linearly in the spine neck. So we have here this example how the voltage is changing with the distance from the North Pole. Again, showing that there is a penetration, large change with the distance much longer than the Debye length when a current is injected. So another interesting aspect from this model is that we can estimate, if we look at the difference of the voltage between the head and the dendrite as a function of the injected current for different radius of the spine neck. From 100, if we diminish to 25, you can see a large deviation from Ohm's law. It's not a straight line, and it depends on this radius of the neck. So we recall the Ohm's law, which says that V head minus V dendrite is proportional to an effective resistance multiplied by the injected current. So this is also amplified here when we looked at the resistance. We plotted this resistance here, estimated from the Poisson-Nernst-Planck equation as a function of the neck radius R0, and the classical wire resistance theory said it is inversely proportional to R0 to the power squared. But here, when we looked at this curve, they could only be fitted with R to some power alpha, and we show here these different power for that is associated with different injected current. And again, the fact that it depends on this injected current is a deviation from the classical theory of conduction. So then we perform another simulation to look, for example, at the influence of the potassium diffusion coefficient. And what was very interesting is that although in the literature there is no absolute consensus about what is this value, and it can vary when you measure this outside the cell, from 2000, where inside the cell, due to tortuosity, the diffusion coefficient can probably be divided by 10. However, if we look, let's say, from 1000 to 500, you can see that changing this diffusion coefficient leads to a voltage in the head, which is almost, in this range, almost constant. Around for this different value here, that for an injected current of 150. So which that even if we make a mistake of, let's say, an error of this number of 10 to, let's say, even 200%, it doesn't have much influence on the final resistance. And I will explain to you why in the case of a wire only, where we can do the exact calculations. So this is obtained again by a numerical simulation of the P&P equations. So now let's focus on, so we have this dendritic spine. It can be divided here into two parts. Let's focus on the case of a wire, which is going to be an approximation of the dendritic spine neck, and let's use this equation to calculate explicitly what's the voltage when a current is injected. So for this, we can use the equation at steady state, which is the P&P equation with three species: potassium, sodium, and ionic species. And we are going to assume here electroneutrality, which again is just it's an assumption, and we're going to see if we push this assumption what it leads to. We have already shown in the literature, I mean the YouTube, we made several YouTube where we explore the case where there's known electron neutrality, and I also invite the interested person to look also in our publications. So this is now the boundary conditions. At the top here, we are going to, so probably I should say, so at the top here, we use this injection flux for sodium only. We assume that there is no potassium injected and no negative charges, while the concentration is maintained here at the base, where the base is supposed to be connected to the rest of the dendrite. So what is interesting here, this has electron neutrality, impose that the negative charges is equal to the positive charges, and this now can be used to solve the equation. So we can get the voltage as a function of the concentration and the concentration explicitly. So there is the concentration due to the injection term, and there is a term here because the equations are nonlinear, which tells us how the potassium and the negative charges are influencing, leading to this correction. And we can see here what is the concentration of sodium as a function of the position for different radius of the neck. And this is a comparison of the simulation versus the analytical solution that we have calculated here. So then what we can do is to estimate the voltage. So the voltage is not a linear function of the injected current. It depends on the log, and it is independent of this potassium concentration. In addition, the resistance again is depends on the log of the injected current, the length of the spine, the concentration of ions, the diffusion coefficient of the sodium, and the Faraday constant. And you have here the result of the voltage as a function of x, the voltage as a function of injected current, and you can see here the deviation, it's not linear, and the most drastic effect is the resistance with respect to the radius r0. And again, it depends on the injected current, and you have here a couple of examples of different injected current, and this is a simulation versus analytical formula that we have used here. And so again, we have a large deviation compared to the classical conduction law, which is that the resistance is inversely proportional to the radius squared. And so all these calculations are summarized in these references published in Physical Chemistry and Chemical Physics last year. So finally, we wanted to study the effect of the geometry, specifically the constriction, the neck constriction, because we have seen, and it is known, that you have a lot of organelles, such as a spine apparatus, that can be present here. So you have a spine apparatus, and that can lead to constriction because particles see a very crowded environment. So we model this using an hyperboloid with a section here which is an ellipse with two parameters, A and B, which characterize the axis of the ellipse. So we cannot anymore approximate this as a wire. So then we ask the question, how does a constriction affect the neck electrical property and the resistance? Probably, and this is the main message here, is that the shortest path has the major consequences on the resistance compared to the rest. But the question was how specifically. That's why we needed to calculate the resistivity with respect to the geometry. So this is the elliptical base, which is a classical ellipse, and then we have here this paraboloid. Then we have already discussed the Poisson-Nernst-Planck equation. And then we have now integrated here, this is a boundary condition that we're going to integrate. So again, we have this connection with the dendrite where we impose a fixed concentration and no flux of negative charges here at x equals L. While, at x equals L, which is here, while we injected here a positive current. So here is the result for the voltage and the resistance. Now we have an even more complicated function of the voltage as a function of the geometrical parameters such as the length, C, which comes into the description of the hyperboloid, AB is the surface, pi AB is the surface of the narrowest ellipse. And then we have here the resistance. How the resistance depends? Again, it depends on the log of the injected current and the different geometrical parameters that are shown here. This is the voltage as a function of the current, the resistance as a function of A, which is the larger axis for the ellipse. And again, this curve here match very well the curves obtained by simulation. And you can see here the effect of the voltage as a function of the distance here, especially at 0.5, where we have the narrow constriction, and you can see the very large effect here, the huge effect for very narrow constriction on the distribution of voltage as a function of the position. This is the axis x from the top to the head. So conclusion, the voltage distribution in spine can be computed from this Poisson-Nernst-Planck equation. The stochastic description from Smolorowski is not appropriate because it's too hard to solve, and probably we are missing here all the fluctuation effect, which could be very important, especially when we don't assume electroneutrality. Ohm's law does not apply for many reasons, and we have seen here what's the deviations from Ohm's law. This said that there are probably a large gradient of concentration, especially when a current is injected, and we should expect a large penetration of the current inside the dendritic spine, which may have a lot of consequences, which to that day have been completely neglected. And finally, the organelle distribution that can lead to narrow passages can also have a drastic effect on the voltage, on the electric field, and ultimately on the voltage relation inside the dendritic spine. So I hope that through this short YouTube, I succeeded to explain what is the consequence of applying this Poisson-Nernst-Planck equation to understand voltage in the dendritic spine. And in the next YouTube, I'll explain how we have used this to extract from voltage dies the effective resistance of a dendritic spine. Thank you for listening.